Get ready to have your socks knocked off. This is going to be a great talk. So it's a great privilege to introduce the keynote speaker, Dr. Feng Zhang. Feng is a professor of brain and cognitive sciences and bioengineering at MIT. He's an investigator of the Howard Hughes Medical Institute. And he's also a member of the Broad Institute. He did his undergraduate degree at Harvard and then came to Stanford for his PhD, where he worked with Karl Dieseroth and developed the optogenetics technology. He, I think, was the best of the best Stanford grad students, so much so that when I first moved to Stanford, I was on the graduate admissions committee, and they have like a form they give us to rank the applicants, like outstanding or good, very good, outstanding, and then it was like the next Feng Zhang, like they had his name there. Then he, after a tremendously successful PhD, he won a junior fellowship from the Harvard Society of Fellows, and here he started to think about ways of engineering the genome, and he started to invent and develop genome engineering technologies, and he is really the quintessence of an engineer scientist and inventing new technologies to answer important biological questions, which he'll share with us. He's won many prizes. He's an elected member of the National Academy of Sciences, the National Academy of Medicine, all of the things you're elected to, he's elected to them. And he's a wonderful, inspiring scientist, and looking forward to hearing the latest.
Okay, great. Thank you. Thank you so much for the embarrassing introduction. It's really a great pleasure to be here. And I really enjoy learning. I don't work on neurodegeneration, but I think coming to this conference and really hearing about all the exciting things that are going on and new understanding and discovery is really very inspiring. So I'm looking forward to learning more and interacting with many of you. So I put together a few things about the projects that we're working on in the lab. And so what I want to tell you about today is some of our continuing work on developing technologies for genetic medicine. I started to work on biological research when I was a high school student. And I had a chance to volunteer in a gene therapy lab at one of the hospitals in Des Moines, Iowa. And it's through that early experience and exposure to biology that I became very interested in and inspired by this idea that you can develop a medicine based on genetic principles and using engineering principles to be able to build things that are modular and have predictable effect when you put into a cell or into the body. So I put this picture here showing a satellite getting delivered into outer space with a rocket because I wanted to kind of highlight this idea of modularity. Genetic medicine is very much like this. It has two components to it. There is a vehicle which can shuttle the genetic payload to the right cells in the right tissue in the body. And then there are the payloads. These are the molecules that if you are able to introduce into the right cells, then they can carry out successful modification or modulation of the disease process. So whether it's correcting genetic mutations or changing the ways that genes are expressed in cells or any number of other things, this is kind of the idea behind gene therapy and genetic medicine. So many groups have done really important pioneering work on developing different aspects of these systems. And over the past decade or so, the payload technologies have really advanced dramatically. But what has lagged behind is the vehicle technology for getting things into the right cells and into the right parts of the body. So in fact, if you go on to the FDA clinical trial website and you look at gene therapy applications, things that are actually in the clinic, they largely focus on 3 different tissues. They target cells in the brain, in the blood, cells in the eye, and also cells in the liver. So we're here, talking about neurodegeneration, and you may ask, why aren't there more gene therapy or genetic medicine trials targeting the brain? And so one of the reasons for that is because we don't have the most powerful and the most versatile delivery systems to get into the nervous system. And also there are many other organs in the body, the kidney, the pancreas, all of the skeletal muscles in the body. We don't have good ways to efficiently and also safely target those tissues either. So what I want to share with you today are then 2 stories that we have been working on that really look at developing new delivery strategies to be able to target different tissues in the body. And I have a little bit of something that we've been working on as a sort of new direction in the lab that I'll tell you about in the end. So the first part concerns proteins that are found in the human genome. These are proteins that were once upon a time, probably on the order of 30 to 50 million years ago, inherited from viruses. And then these genes have been co-opted by our ancestors, evolutionary ancestors, and then they became a part of the human genome. They are expressed, they do important things in the body, and they still form virus-like structures. And so we have been studying them and trying to engineer these human proteins to become delivery vehicles. And then the second story talks about systems found in bacteria. Just like these human proteins that were inherited or exapted from viruses, bacteria have also borrowed things from bacterial viruses. And so the second story talks about injection systems that bacteria have adapted from their viral infections and then turn them into things that can deliver things to other cells. So let's look at the first one. So since the human genome was first sequenced, bioinformaticians have been analyzing genes in the human genome. So there are about 20,000 protein coding genes. And then by looking through all these different genes and looking at sequence homology to retroviral structural proteins, GAG, what people found is that, especially Campilla and colleagues, they found that there are about 80 genes in the human genome that share strong homology to the retroviral GAG protein. And they then classified these into different groups. The sushi family are one set of human genes that are thought to derive themselves from the sushi retrotransposon, which are LTR transposons that form capsids. And then there are the scan family and also the paraneoplastic family. And there's this other gene that's expressed in the brain called ARC, involved in immediate early gene expression and also synaptic plasticity. These are all things that have very strong resemblance to the retroviral structural GAG protein. And if you look at the domain annotation, you'll see that most of them contain these blue regions that are sort of resembling the capsid domain of the GAG protein. And so a few years ago, both Jason Shepard's group and also Travis Thompson's group showed that the ARC protein, which is expressed in the brain and is found in anywhere from insects all the way to mammals, these remain capable of forming viral capsids. And more importantly, in Drosophila, the Drosophila version of ARC protein can package RNA and also can deliver this RNA across the neuromuscular junction. So it's a very interesting kind of system. So we became very interested in these systems after reading these papers. We thought that if in the endogenous proteome there are these virus structure-like things, maybe we can co-opt them, turn them into systems to deliver therapeutic proteins or RNA to cells to be able to treat disease. So 3 of my lab members, Vicky, Yugang, and also Ramya, they began to study one of these families of proteins from the human genome, and that's the paraneoplastic MA antigen family, so PNMA. And there are many different PNMAs in the human genome. They are all homologously related to GAG, and many of them carry these blue N-terminal and also C-terminal capsid domains that are found in retroviral capsid proteins. And so we thought to study what these proteins do. So one of the first things that we did is that we synthesized all these different genes. We recodon optimized so that if there are any cryptic regulatory sequences, we got rid of them. And then we introduced them into human cells. And so most of them are able to express, and they can form virus-like structures, and many of them are even secreted by the cells. But one of these PNMAs turned out to be different from the rest, and that's PNMA2. So these are EM images. So on the bottom, you can see the particles of PNMA2 that are released by human cells that express this PNMA2 protein. The way that they are released is different from the way that retroviruses or these other PNMA2s are released. They are released as protein-only capsids. They don't have a lipid membrane that kind of envelope the capsid on the inside. So this is what these capsids look like. They actually look like other protein viruses like AAV particles, which also don't have a lipid membrane. So we thought, let's study these particles to see what do they package and what do they do. So one of the first things is we purified a lot of them, and then we extracted whether it's RNA or DNA that's inside of these capsids. And interestingly, we found that the capsid doesn't really package very much of anything. So here, this is the VLP, so virus-like particle fraction. So these are the PNMA2 particles. We extracted it, we extracted the RNA, and then we did RNA-seq. And we found that really nothing is enriched in this RNA fraction. And so that was very interesting. So we were a little perplexed because this is different from the ARC protein and also from another protein called PEG10 that we studied. Those proteins, they form capsids and they package their own mRNA. But PNMA2 doesn't package mRNA. So we then carried out biochemical studies of how this particle works. And so we found that these proteins express very well. If you heterologously introduce them into E. coli, you can purify large quantities of recombinant protein. And these proteins are well behaved. They form virus-like structures right in bacteria. And so here are EM images showing what these sort of recombinant virus capsids look like, or PNMA2 capsids look like. If you remove salt, if you just dialyze out salt from the buffer, these capsids will fall apart. They'll disassemble into monomeric protein. And if you add salt and also calcium back to the solution, they will then reform these virus-like structures. So it's a very nice, well-behaved biochemical system. They can form, you can open them up, and then you can reform them by just changing the salt concentration. We thought this is nice because if you can open it up and put it back together, this is almost like a container that you can open up and stuff things into it and then close it back together. So we thought maybe we can engineer this to get it to package things of use. So to do that, we first solved the cryo-EM structure. This is a well-behaved protein, so it's actually fairly easy to solve the structure. And this is what the structure looks like. Here I'm showing you the cross-section of this capsid. So you're looking at the interior surface of this capsid. Red is negative charge, and then blue is positive charge. And so you see that the inside is quite negative. And this makes sense, in the sense that we don't see any mRNA getting loaded inside. And that's because mRNA is negatively charged. And if you have a negative charged surface on the inside, then they will repel each other. You're not going to get any packaging. But this also gave us the idea of how to engineer this to turn it into something that can package mRNA for delivery. And so what we did is we thought, let's flip the charge on the inside. So we looked at the wild-type protein, which has a region that's on the C-terminus that is negatively charged. And we thought, let's chop this off and replace it with a fragment that is positively charged. And we took a domain from the CCMV, which is a plant virus that is well characterized for binding to RNA. So it's a very positive charged domain. So we call this engineered PNMA2. And what we found is that this particular protein is also very well behaved. but it only packages or it only forms these capsids in the presence of RNA. So in a way, you have to have charge balance. So you have to have positive charge and also negative charge together in this capsid in order for it to even form. So this is nice. We thought now we can package mRNA, so maybe we can use it to deliver mRNA into cells for different applications. And so this is an early attempt at doing that. So what we did is that we used the engineered PMA2 and we packaged the mRNA for the Cre recombinase protein. And we'll have a reporter cell line that has a flox GFP so that if Cre is expressed, then it would activate GFP expression. And so what we found is that over here in our experimental condition, with Cre mRNA packaged inside of these engineered PMA2, just to make sure that we are delivering things inside of the capsid, we also treat the capsids with RNAs, just so that we get rid of any RNA that's on the outside that didn't get packaged. And we saw that we can get GFP activation in the recipient cells. And as a control, we just directly transfected the Cre mRNA into cells, and you can also get GFP activation. And so this is something that shows that we can use this general approach where you can recombinantly purify these capsid-forming protein in a biochemical, well-controlled environment, mix it with the payload you're trying to load, and they will form capsids, and then you can deliver it into cells. And so in contrast to cellular-based production systems for particles, this is much cleaner. Because if those of you who make AAV, you often see that when you use cells to produce AAV, number one, you produce empty particles. So these are just viral protein that don't have anything, any of the payload loaded inside. And then second, they usually secrete a lot of other junk, a lot of things that if you concentrate and put onto cells, that can cause toxicity and also immunogenic reactions in the animals that you inject into. And so we're now continuing this approach to try to get the system to work better. And so the problem that we're trying to solve now has to do with the efficiency of endosomal escape. Because the way that these particles work is that they will bind to the cell surface, it will get endocytosed into the cell and trapped inside the endosomal compartment. They will traffic from early to mid to late endosome, and through that process, they have to somehow come out. And it doesn't come out very well right now. It's similar to what people can get with lipid nanoparticles, which is about 1% of escape efficiency. So we're trying to figure out how to get these particles to escape at a higher level of efficiency. So that's what we're working on now. And so because of that problem, that inspired us to then also think about the second approach that we've been working on. What if we can bypass endosomal escape? So rather than going through these endosomal transport processes, just go through the cytoplasmic membrane and unload into the middle of the cell. So this is what Joe, who's a graduate student, has been working on. And these are the prokaryotic injection systems. They don't go through endocytosis. Instead, they bind to the cell membrane and they physically puncture the cell membrane to then unload something into the cytoplasm. So let me tell you a bit about how this works. So this is a schematic showing how these injection systems work. They are made by bacteria. They are secreted by bacteria, so they're soluble injection systems. And they look very much like a syringe, a nanoscopic microsyringe. Over here on the right is the cryo-EM structure that's solved by Zhang and colleagues at Beijing University. And you can see that they look very cool. They have a long shaft, and they have a hollow core on the inside. This is where the payload can get loaded. Usually it's some type of bacterial toxin or effector protein that the bacteria wants to get into their host cell. What is really cool is that they have this really sharp spike at the end of it. And this spike is what gets physically pushed through the cell membrane to then unload things into the middle of the cell. And it also has these orange things, which are called tail fibers. They are the attachment anchors. They can bind to the cell surface based on receptor-mediated recognition of whatever ligand that's on the cell surface so that they can dock on. And so basically it works like this. So you have a syringe. these tail fibers will unfold and bind to the cell surface, dock onto the ligand. And then upon recognition, it will trigger this spring-loaded mechanism to then compress. When it compresses, this sharp spike protein gets pushed through the cell membrane, and then the payload can get unloaded into the cytoplasm. So we thought this is a cool system. It will bypass endosomal transport, and we can use this to deliver useful therapeutic molecules. So we borrowed the entire gene locus for these syringes from a bacteria called Photorhabdis, and we transported it into E. coli so that it's easier to work with. And using E. coli, we found that we can produce these syringes. And these are EM images showing what the syringes look like. They're long and narrow, and they package payloads in the middle. The way that a payload protein gets loaded is that they have a signal peptide. And so as long as you take the signal peptide from the native effector proteins, and if you put it onto any other protein you want to load into it, it will also get loaded in. So that's how we produce these things. So with this, we established that we can use E. coli to produce them, and then we had to verify that they work. So here is an experiment where these syringes, they are naturally made by Photorhabdis, and Photorhabdis use these syringes to target the gut lining cells of the insect that they live in. And so they have natural insect cell recognition capability, but they don't recognize mammalian cells. So we had to test the function. What we did is that we took the SF9 insect cells, and then we applied these E. coli-produced syringes onto these insect cells to see whether or not the syringes recognize insect cells and they inject their toxin into the insect cell to then kill the insect cell. So we found that this worked. So with the wild-type system, so unmodified syringes with the toxin loaded inside, we get quite a bit of killing of the SF9 insect cells. But then if we delete the payload, so if there's no toxin, if we delete the payload loaders, then nothing gets loaded. If we get rid of the tail fiber, none of those sort of produce the same level of toxicity. And so this shows that we are able to produce functional syringes, and they inject the toxin in a binding and also injection-dependent way. And so this is something that allowed us to get some confidence that we can make functional syringes. So then we had to figure out how to reprogram it, because these are naturally insect cell recognizing, but we want them to recognize mammalian cells. And so to do that, we looked at the tail fiber protein, and we thought that if this is what's allowing it to recognize a target cell, maybe we can modify the binding domain, then it will recognize sort of other types of cells. So we started with the wild type tail fiber, and my graduate student who was working on it tried probably more than 100 different designs of how to remove the binding domain and fusing a new binding domain to it, and all of them failed. And part of that is because we didn't have a structural understanding of how these tail fibers work. And so just about we were ready to give up, AlphaFold 2 came out. By the way, we tried to solve the structure, but it was hard to solve because it's very sort of flimsy. So it's hard to get a really clear idea of what it looks like. And so with AlphaFold 2, we were able to get a predicted structure. And so based on that prediction, we then designed a few more sort of modifications, and these worked. And so here we were able to make a cut right there and get rid of the binding domain, and we can now replace it with either a binding domain from the human adenovirus 5, which recognizes a number of different human cell types, and also we were able to replace it with a DARPin, which is a programmable protein binding domain that we can use it to recognize different things. So we have AD5 and also a EGFR targeting DARPin. And then as a control, we also used a DARPin that recognizes an intracellular protein rather than something that's on the cell surface. So that shouldn't work. So again, we used the reporter cell line. We have now a human lung cancer cell line, A549. And then this is both EGFR positive and also recognizable by AD5. So we are delivering Cre recombinase now to activate a reporter that's in these cells. So with the wild-type tail fiber, there's really no signal. And also with the truncated tail fiber, there's no signal, because this only recognizes insect cells. It wouldn't recognize a human lung cancer cell. So then what we found is that with AD5 fusion, we can now activate the reporter expression. And then if we mutate the AD5 domain so that it doesn't bind anymore, there's really no activity. And then the EGFR DARPin is able to activate expression, and then the lysozyme doesn't really activate expression. So this shows that we can now reprogram the system to recognize a mammalian cell rather than an insect cell. And this is really sort of with the help of AlphaFold 2, which allowed us to get some understanding of what the structure looks like. So then we also tested whether or not we can deliver more useful proteins. We put the signal peptide onto either Cas9 or onto a zinc finger deaminase, and we found that you can deliver these proteins to be able to achieve gene editing in the recipient cells. So this shows that we can also deliver more useful molecules. And then we wanted to see how well does this system reprogram. Can we get it to target a variety of different proteins on the cell surface? So to do this, we engineered different tail fibers that have different protein tags on this tail fiber. So we simply cut off the binding domain and we fuse HA or flag or moon tag, etc., onto the tail fiber. And then we also generated a number of different cell lines that have antibodies able to recognize these different peptides. And so on the vertical axis, we have different cell lines that express anti-HA or anti-flag and so forth. So with this, what we found is that you can get very good reprogramming specificity where the protein peptide that's on the tail fiber has to match the antibody that's on the cell surface. And if they don't match, then you don't get very efficient delivery into those cells. And so this shows that the tail fiber is a modular domain. You can cut off the binding domain from it and then attach different things onto the structural part of the tail fiber to get this syringe to be able to inject into different types of cells. So as long as you can find a good binder, then you can get it to target those cells. We also tested whether or not the system might work in vivo. So we did a stereotactic injection into the hippocampus. So here we used the AD5 binding domain, and then we injected Cre recombinase into an M-Cherry flox mouse. And so what we saw is that you can get activation of M-Cherry expression in sort of dentate gyrus neuronal cells in these injected animals. So the efficacy is not perfect. So what we're seeing here is sort of modest distribution. And so we're now working on trying to get the system to work to diffuse better. And part of that has to do with changing the size of these syringes. So we don't want really long shafts. We want it to be short so that it can more freely diffuse and be able to get to farther distances from the injection site. So that's something that we're working on now. Another thing that we're working on is that ideally we want to be able to deliver things that are not just protein, but other things, nucleic acids or protein RNA complexes like RNP for gene editing. To do that, we initially tried to engineer the shaft so that we can deliver things into it. But if you recall, the geometry of the shaft is very long and narrow. And so when a protein gets loaded inside, it has to be at least partially unfolded in order to get loaded inside. And so instead of, and if you want to load more complicated things, unfolding it is not always a possibility. So then we thought maybe we can come up with a different strategy to deliver things that are more complicated. And so we then turned ourselves to nature for inspiration. And so it turns out that a type 6 secretion system, by the way, there are many different types of secretion systems in bacteria, type 1, type 2, type 3, type 4, etc. And these syringes actually derive from one of these secretion systems. And so the type 6 secretion system, rather than loading the payload, which I'm showing here in red, rather than loading that inside of the shaft of the injection system, that payload is covalently linked to the tip, to the spike of that injection system. So then it's basically like, the spike is basically like a cannonball. It shoots through the cell membrane, and then whatever that's attached to it also gets dragged into the cell. So we thought maybe we can take this strategy and then fuse our payload, more sophisticated payload, onto the spike protein, so that when the spike is pushed to the cell membrane, it literally drags the payload into the cell as well. So that's what we try to do. So PVC10 and PVC8 are proteins that form this sharp spike. So we thought if we covalently fuse a payload to either PVC10 or PVC8, then we'll be able to get it attached onto the tip of the spike, and then you can get it injected into the cell. So that's what we thought to accomplish. You can either make one fusion, or you can make two fusion, or you can make four proteins fused to the tip, so you can deliver different amounts to the cell. So this is basically the idea. If we have this fused to it, then when this goes through the cell membrane, it will drag the payload right into the cell. So that's what we're trying to do. So it turns out that this can work. So here are EM images of the engineered PVCs, the syringes, with the payload attached to the tip. And what you see is that we're attaching Cas9 to the tip, and you can see the Cas9 particles. This is one molecule Cas9 on the tip. Here are two molecules of Cas9 on the tip. And so you can literally attach it to the tip of these syringes. It's very cool. And we have also gotten better at forming consistent length or uniform length shafts, which helps with delivery as well. So then we found that we can get the system to successfully deliver Cas9 guide RNA RNP complexes into the cell. And so these are sort of experiments where we injected Cas9 fused to PVC10, and then this has the guide RNA already bound to it so that it can deliver the whole gene editing machinery directly into the cell. So this is something that we're continuing to work on to get efficiency to be better, and also in addition to delivering Cas9, we also just want to load mRNA. And so we're also working on ways to be able to sort of chemically, using click chemistry or other conjugation methods, to then link mRNA onto these spike to drag that into the cell. So the two stories that I shared with you today are two things that we have been learning from nature. So whether it's endogenous human capsid proteins or these bacterial sort of viral-derived injection systems, they came from nature's diversity. And so nature really has created almost anything that we could possibly imagine. And so we're continuing to do that. And so I want to show you this graph, which is somewhat out of date now, but it shows the amount of biodiversity sequencing information that has been collected over the years. And this is an exponentially growing trend. And so to date, if you look at all of the protein sequences in databases, they aggregate to be almost 8 billion different unique proteins. And so out of all that, that's a lot of really sort of rich diversity that we can mine from. Most of them we actually don't know what they do. And so if we continue to try to mine from there, either using human intelligence or artificial intelligence or a combination of the two, I think we're going to be able to discover many more interesting biological processes, and also some of them will certainly become useful therapeutic strategies as well. So that's one part of what we're working on. And then on the second part of the lab, we are kind of shifting gears and trying to think about how to engineer biological resilience. And so I want to tell you about one story that we've been working on, just a little bit of it. So this has to do with the immune system. And so a lot of talks that I learned from today touch on the immune system. And so as we get older and older, our immune system doesn't work as well. And the immune system is very important. And so the way the immune system works is that when we're young, we are starting to build our immune system to really develop a robust sort of set of immunological capabilities. And then in adulthood, our immune system is very good. It protects us from infections. It sort of reduces the risk for developing cancer. But then as we get older, the immune system starts to get tired. And so this aging immune system starts to decline. And then this is when we start to have infections, cancer, and also many other sort of serious illnesses. So we thought, can we figure out a way to prolong this period of homeostasis when the immune system is strong? So one of the things that's really important for immunological development is the thymus. And so the thymus is something that is changing in our body, and it's actually one of the organs that begins to degenerate quite early on, beginning at the onset of puberty. And so we analyzed thymus. So these are some images showing you how thymus degenerates. This is the thymus of a 3-month-old. You can see it's very healthy, a lot of thymoepithelial cells in blue. By the time a person is 17 years old, you can see that there's dramatic decline in these thymoepithelial cells. And by the time a person is 70 years old, there's really nothing left. And so this is why, this is how the thymus declines. The thymus is very important for generating new T cells. And so as the thymal tissue degenerates, our ability to make new T cells also declines. And so one thing that we thought is, can we figure out a way to regenerate the thymus, or to at least sort of substitute for the thymic function in old age? So Mirko is one of the postdocs in the lab, and we began to look at what changes as the thymus gets older and older. And so what we did is we profiled thymic tissue, and then what we found is that there are there are many factors that change as the thymus gets older, but there are 3 of the factors that seem to be quite important for especially T cell development. And so we thought, okay, maybe we can take some of these thymic factors that are declining in old age and use lipid nanoparticles and mRNA technology to deliver it into the body so that we can replenish it. Lipid nanoparticle doesn't target the thymus. In fact, very little goes to the thymus, but they go into the liver very well. So they get into hepatocytes. And it turns out that the liver actually has some important roles in T cell development and in embryonic development, or during embryonic development. But then it's also a place where your entire blood flow passes through, even in adulthood. And so we thought, if we can express some of these thymic factors in the liver, then when blood flows through, progenitor cells or other cells will still get exposed to these factors that are normally in the thymus, and maybe we can provide the same function the thymus is doing. And so in a way, we're trying to temporarily use the liver to sort of substitute for some of the lost thymic function. So that's what we're trying to do here. We're hoping that by introducing these sort of declining thymic factors, we can then get the T cells to develop better. So we generated mRNA for these 3 factors, so DLL1, FLT3, and also IL-7, and then we package them up using lipid nanoparticles to deliver it in vivo. And what we find is that we can get pretty good expression of all 3 of these different factors using mRNA delivery, which is a good thing. So then we injected these into mice. So what we did is that we injected over the course of 3 weeks using 8 doses of the mRNA, just so that we can have enough of these factors expressed in the liver. We did compare with injection of the recombinant protein. So rather than delivering mRNA, we just injected the protein. What we found is that the protein delivery doesn't work as well. And also it causes more toxicity. And because a lot of these cytokines, if you deliver systemically, it's much more toxic than if you just locally express it in a specific tissue. So here is an experiment where we did a peptide vaccine challenge. And so what happens over aging is that the TCR repertoire declines. The diversity of that TCR repertoire declines. And so as the immune system gets older and older, the response to a new vaccine becomes much weaker because there's less TCR diversity to draw from. And so what we did here is that if you look at these white dots here, these are wild-type mice, untreated, of different age. And then we immunized these mice with a peptide vaccine. And so what we find is you can measure the amount of sort of immune response in these in these mice. And you can see that they follow a very nice correlation from young to old age. And so you can basically generate a fitted curve here, and then you can then measure the vaccination response of any animal, and you can plot that vaccination response on this curve to then be able to estimate the biological age of the animal. And so then what we did is that we took either a control luciferase mRNA treated mouse or mice that have been treated with our 3 thymic factors. And what we find is that the vaccination response of mice treated with the thymic factors is much stronger than the same age mouse. And in fact, it kind of rejuvenates by about 24 weeks of age. And so this shows that we can sort of boost the immunological resilience of the system by introducing these 3 different factors into mice. We found that the same treatment also improves sort of checkpoint blockade immunotherapy response. And so here we have mice where we delivered using LNP the 3 factors over 8 different doses over 3 weeks. And then also we then sort of injected tumor cells into these animals. And what we found is that by giving anti-PD-1 or PD-L1 sort of immunotherapy, the treated mice had a much better response for the tumor. And so the tumor growth rate dramatically declined compared to animals treated with the luciferase control mRNA. And so this is something that another way of looking at our perturbation on the immunological resilience. And then we look at survival. So when you treat with the thymic factors, because the tumors don't grow as fast, the survival is also much longer than if you treat it with just the control luciferase mRNA. And then, so that's what we're continuing to work on. It's still a work in progress, but we're pretty excited about engineering these sort of physiological systems by either reconstituting functions that are lost or introducing synthetic functions. We thought this is one way that we can begin to have a more sort of robust modulation of health within an animal. Okay, last but not least, I just want to acknowledge my team. This is my lab at MIT, and then our support, and also if you have any questions or are interested in joining us, feel free to send me an email. Thank you again. It's really an honor to be here.
Thanks so much, Fang. We have plenty of time for discussion.
Great.
Hi. Thank you. It was a mesmerizing talk. I have 2 questions. First one relates to the injection of the cargo that you presented. So if you're tying your cargo to essentially the needle that's there, the rest of that structure is essentially just there to facilitate the introduction of that needle into the cytoplasm, because you're no longer using that tube to carry anything. How well do you understand the mechanism of that transfer, and are there, I guess, homologous structures that can facilitate that instead of using the tube structure, I guess, that you started with?
Yeah, that's a really good question. So thank you for asking that. The syringe, one of the pictures that I showed, you saw that there's some variability in the length of the shaft. So on one of the EM images, you see there's quite a bit of diversity. This one, right? So some are long, and some are shorter, and so you're exactly right. So the syringe we're not using it anymore if we're just tethering on the outside, but we do need a syringe to have some length there, because it's a spring-loaded mechanism, and that the spring-loaded power is stored in the length of the shaft. And so if you make it too short, there's not going to be enough to then be able to push it through the cell membrane. But we can keep it as minimal as possible. And so the way that you can do that is that the length of the shaft is determined by a protein called the tape measure protein. So what people have shown is that these tape measure proteins in phages, they're in modular domains. So depending on the number of domains that you repeat in this tape measure protein, you can change the length of the phage tail, which is the shaft on a sort of bacterial phage. And so that's something that we haven't yet explored, but I think you can use to be able to better control the length of the shaft. You can keep it to the most minimal tape measure domain, and then that can keep it minimally but still active.
That's beautiful. The second question is the reversal, so that thymic effect, how late can you go to reverse it? So you showed, for example, the reversal of thymic function from 24 weeks to 12.
Right.
How far out can you go?
The oldest animal that we have tested this in is about 70 weeks. So that's kind of the late middle age. We haven't had enough animals older than that to be able to test and be able to have statistical power. So we don't know. But at 70 weeks, it still works.
Cool. Thank you.
I have a question.
Yeah.
I have a question. I think you partially answered to it. I was just wondering how long could you use these these viral particles for treating the animals? How long could you use them before the immune system recognizes them and starts clearing them out?
Yeah, so there are two delivery systems I talked about. The first one is endogenous protein-based. For those systems, I think they are less immunogenic because you do find them circulating in the body. The second one, which is bacterial derived, that's probably going to be more immunogenic. And we have done injections to the brain, which didn't show immunogenicity, but that's not a really good measure. Because the brain is somewhat of a immune privileged area. But I think repeated injections systemically will probably be immunogenic. So we'll have to work on trying to remove some of the most antigenic parts of that syringe.
That's cool. And just one final question. Would you consider modifying these viral proteins to overcome the immune system in somehow? Do you have ideas on how to do it?
Yeah, there are ways to do that. So usually they're done by first running either computational or experimental analysis to see what are the most MHC1 and also MHC2 reactive components on this protein. So you can basically make peptide libraries of all the proteins in the syringe, run it on the MHC1, MHC2 panel, and see what are the most reactive peptides. Then you can go in and based on structure to alter those peptides. You can change it. You can make amino acid substitutions that preserve the same structure. You can do this computationally now, especially with some of the more advanced sort of protein design tools. So that's how you would go about doing that. But you still have to do a lot of empirical testing. You probably have to make hundreds of different versions of this and then test it to see which ones reduce immunogenicity. It certainly works. People have done this with antibodies. We have done this with Cas9. And I think the syringe is a more complicated machine, so it's going to be more work. But I think that general approach will probably work.
Thank you.
Yeah.
Hi. Beautiful talk. And as someone who thinks a lot about drug delivery to the brain, I found this really fascinating. And I was just curious, since we're all here at a neurodegeneration conference and thinking about drug delivery to the brain, I would love to get your thoughts on how you would potentially utilize these particles for brain delivery that may be less invasive than what you showed in terms of the direct injection, right? I could foresee some challenges of getting across, say, the blood-brain barrier. So I don't know if that's something you've thought about.
Yeah, so with these endogenous particles, what we are trying now is try to functionalize the outside of these particles with targeting domains. So we're now exploring what other people have found to be somewhat successful, which is anti-transferrin receptor antibody, and trying to use that to cross the BBB. And so that's something we're trying now. We don't know how it will work yet.
Yeah.
But yeah.
Then maybe a quick question following that. How would you prevent it from unloading the payload in the first cell type that it sees, which would be the endothelial cells that have the transferrin receptors?
So transcytosis is different from endocytosis.
Right.
What we found with these GAG-like particles is that they assemble and disassemble in a pH-dependent way. So it's only when the pH drops by the time you get to mid to late endosome that the particle falls apart and allow the mRNA to come outside.
Got it. Thank you.
Yeah.
Yeah, no, that's really exciting, and yeah, especially also the rejuvenating the immune system makes a ton of sense and is exciting. But to the extent that thymic education during development is sort of trying to restrict the immune system and guide self and non-self, and then
right.
Ideally as we get older and wiser, our immune system needs less of that. I mean, is there a concern, and can you sort of quantify if you make the liver into a thymus? Because the thymus, in theory, I think, and especially the epithelium, is especially good at presenting self and whatnot. Have you looked, and can you tell if you might run into trouble at the other extreme where you...
Are you asking whether or not we might see increased autoimmunity?
Exactly.
Yeah, if we do a treatment like this. Yeah, that's a great question. So we have done some preliminary testing. So we have done sort of the NOD mice, and which develops autoimmune reactivity. And when we treat a 50 to 60 week old NOD mice, we don't see increase in sort of autoimmunity in that model. But that's just one model. I think we have to do more testing, but so far it looks okay.
To what extent can you, I know that you've kind of relied on nature to give you the particles to start with, but now that you've seen what's out there and how it works, to what extent can you engineer proteins de novo, say, to increase the cavity size or change the constituents within the cavity to bring in, for example, to put binding sequences that would allow certain proteins to be charged in there?
Yeah, that would be amazing to do. I think the long-term holy grail would certainly be to do that, to really just de novo computational design whatever nanostructure you want with the dimension that you desire. So David Baker has done that pretty successfully to generate these nano cages of different sizes. But right now, the computationally designed systems suffer from one challenge, which is that these particles, they don't open up. It's hard to engineer these pH-dependent assembly and so disassembly properties into the de novo design particles. I'm sure eventually we'll get there, but as of right now, we don't have as good of a control for those things yet. So then for these natural particles, what you will see is that the natural diversity of these particles do form capsids of different sizes. PNMA2 is on the smaller side. It's about 23 to 24 nanometers in diameter, so that's very small. The packaging capacity is probably around 3 kilobases of mRNA. But then there are other proteins that do form larger. So PEG10, for example, is about 80 nanometers, or 70 to 80 nanometers in diameter. So that will package a lot more. So what we could do for now as a temporary bridge is to just develop a range of these different particles so that we have different sizes.
Frank, a simple question to the vaccine. The vaccine itself, was that already maxed out, or did you benchmark against vaccine formulations that could also be enhanced in functionality, like with targeting innate immune mechanisms at the same time, etc.? Because there's big differences if you take the same antigen in different formulations.
Right.
Have you looked at that?
We haven't looked at anything more sophisticated like that. We just did a very simple ovalbumin peptide injection, which is a kind of I think a reasonable first test at immune vaccination. Yeah.
Hi, beautiful talk. I'm intrigued by the resilience piece, and I remember people were doing like parabiosis experiments, hooking up young and old mice. I'm curious if there's any overlap of your thymic factors with anything that's been identified in those studies.
Oh, that's a good question. I don't know. Maybe someone from the White Squirrel lab or someone who has done that would recognize these factors. I don't know. That's a really good question.
Thank you.
Thank you very much for the very interesting talk. It has, especially the first part, has my imagination running wild at the moment. I was wondering, when it comes to those molecular syringes, basically jamming through the membrane, regarding amyloidogenesis is often linked to disruptions in membranes and disruption of membranes. Do you know if repeated, basically, injection through the membrane can cause this kind of disruption that maybe even makes protein aggregation worse in the initial case? Or do you have any preliminary data on how potentially the injection could cause disruption of membranes or changes in membrane structure overall?
Yeah, I guess the short answer is I don't know because we haven't tested it. But I don't think it would, because the dose of what you would deliver into the body is probably not going to overwhelm the cells. So maybe you would have 5 syringes per cell at the highest level, and if that's the case, 5 different little holes on the membrane very temporarily will probably get repaired because the membrane is very dynamic. It ruptures and reforms constantly. So I don't think that would be at a level that will cause problems, at least not through that mechanism. Yeah, Hideyuki.
Yeah, okay, thank you. Thank you, very wonderful talk as always. And so if you want to tether to the particular type of cells, so you can make some chimera with a specific, extracellular domain of the protein. Have you tested? And also to make chimeric proteins and to enhance its efficiency, maybe deep mutation scanning like that you did in Cas13 could work. How do you think?
We have done some targeting work. So we usually just use antibodies and we conjugate that to the surface of these capsids or to the tail fiber protein. And so that requires having a good antibody and also a good antigen on the cell surface that is at a relatively high abundance. So for targeting T cells, for example, we can use CD5 antibody, we can use CD3, CD4, CD8 targeting antibody. That will all work. For hematopoietic stem cells, we can use CD117 to target, and we're trying other ones. I think hard to know. I think some of these things we'll have to just do the experiment before we know whether or not it works.
In order to make the chimeric protein to work it better, so have you done the deep mutation scanning?
We haven't done that yet. Yeah,
could work, right?
Yeah.
Absolutely fascinating stuff. I'm curious, so you mentioned that you can engineer these, I'm going to call them legs, because I don't know a better term for these molecular syringes to be more specific to specific molecules on the surface of the cell. Could you use that to make them specific to a subcellular region? So, for example, could you get it to inject only in the postsynapse or something, and then use that to deliver like an mRNA for local translation, or perhaps a specific protein within a specific region of a neuron?
Yeah, that would be really cool. I guess I don't know. We'll have to try, but I guess there are postsynaptic density proteins that are unique to subcellular regions, and you might be able to, develop an antibody against that works well. But I think we'll have to see how it works. Yeah, have to try it. Yeah.
Really great talk, Phil. I was wondering whether you have looked at nuclear delivery of these payloads, the efficiency of that, especially when it comes to engineered PNM2.
Yeah, good question. We don't have nuclear delivery, unfortunately, because if it's through endocytosis, they will get unloaded into the cytoplasm. Although we can deliver RNPs, and if you attach a nuclear localization signal, And if the protein is sufficiently negatively charged, positively charged, it will get into the nucleus. So Cas9 RNP works in that way. But without that, I don't think we'll have any nuclear targeting.
Thank you.
Okay, I think everyone's asked one question. So,
all right.
amazing talk, a lot of inspiring visions, and we now have a party with cheese and wine, and then later tonight, go to the poster session.